in mammals such as human and mice, and total number was turned out to be 45 at the gene level. So the protein level, this number could be twice more. Using molecular cell biology and molecular genetics, we are studying the structure and function of the motors in cellular level and also in vivo level. And we are also studying the mechanism of motility of the motor by hydrolyzing ATP along microtubules using cryo-EM combined with X-ray crystallography and single molecule biophysics. But I don't go in detail today. So today, I'll focus on the three motors related to disease. But molecular genetics opened up a quite exciting area. And we know that, for example, KIF3 control left-right determination of our body, or KIF3 suppress tumor genesis by transporting beta-catenin, L-catenin from cell center to plasma membrane. And KIF4 regulate the PARP1 activity and regulate the activity-dependent neuronal survival and neuronal death. And also KIF26A control enteric nervous system development as a factor of the signaling, GDNF and RET signaling cascade. So the motor proteins is doing very important and interesting work. So this is a microtubule. Let me point out one thing. Microtubule has a polarity, plus end and minus end. So actually the plus end beta tubulin is exposed, and so motor proteins reading this polarity and determining the direction of transport. So in the axon, plus end, microtubule's plus end is pointing distal, apical epithelial cells, the minus end is pointing apical, and this is a fibroblast. So this is sort of the summary of the mechanism of transport. We identify many cases and show that they transport different cargos such as mitochondria, synaptic vesicle protein in axon, and in dendrite, messenger RNA complex, and also receptors. And so this, for example, KIF17 control higher brain functions such as memory and learning. Very exciting. So today, I talk about this KIFs and related disease. First member is the KIF5A. KIF5A is essential for GABA A receptor transport, and KIF5A deletion causes epilepsy. So KIF5 has, subfamily have three members, A, B, C, and they have a redundant function, the redundant cargos, but we want to know the specific function of KIF5A. And this region is specific for KIF5A. So we generated knockout mice, specifically knocked out KIF5A in brain. Conditional, we generated conditional knockout. And so, and the mice was born and looks healthy at a glimpse. As shown here, histology, we didn't saw the difference between genotypes, but we found, we observed, we kept these mice for a long time and observed their behavior, and we found that they have epileptic seizures. So I show you one example. The conditional knockout locomotive state. So actually, you can see the seizure here. A tonic cramp was observed in the hindlimb. Okay, I repeat this again. Okay, like this. Then we studied the inhibitory synaptic transmission system. As you can see here, miniature inhibitory postsynaptic currents changed in conditional knockout. Amplitude is much less, okay? And using hippocampal slices, we studied electrophysiology, and we found iPSCs are affected while AMPA receptor-mediated currents are not changed. So inhibitory system is affected. So then further, we found epileptic form activity in response to electrical stimulation, like in conditional knockout. You can see the seizure, epileptic form activities there. So we thought this could relate to the GABA inhibitory system. So what's wrong? We studied the receptors, GABA receptor, and found surface exposed GABA receptor was decreasing in amount like this in knockout. Okay, so this is the dendrite of the neuron, hippocampal neuron, stained with anti-GABA receptor from the surface. As you can see clearly, the surface exposed GABA receptor was decreased in conditional knockout mice. Interestingly, however, in knockout, the intracellular amount of GABA receptor was increased. That means the receptor cannot be transported from cell center to plasma membrane, accumulate in the cell, inside the cell. And other proteins are not changed. So this slide summarizes the localization of a GABA receptor in wild-type dendrite neuron, and this is knockout, and this is the rescue by expressing K5A, and as you can see, in knockout, the density of the cluster and the size was decreased, as shown here. And that can be rescued by application or expression of K5A, but cannot be rescued by other members, such as K5B, K5C. And this is the knockdown experiment, and that supports this conclusion. Then we try to find out the mechanism of transport of GABA receptor by K5A. So we performed East2 hybrid to find out the binding partner of the K5A tail domain. We picked up GABA receptor associated protein. Okay? So this interaction was confirmed by in vivo immunoprecipitation. As you can see here, and the GABA antibodies picked up the immunoprecipitate 5A, GABA receptor, and GABA. And we can have a reverse data too. So the slides show the co-localization of the K5A and the GABA in the dendrite. Okay. So, and we studied the distribution of GABA also. This is a wild-type neuron dendrite and knockout neuron. And this is the statistics. The GABA is a small molecule and can diffuse at the proximal region. So at the proximal region, there is a not so big change. But at the distal region, the GABA should be actively transported. So in knockout, because K5A is absent, then there is a difference between wild type and knockout in terms of the distribution of GABA. Okay, and we analyzed the dynamics transport of GABA receptors in wild type and knockout. And we found there are 4 types movement, stationary, vibrating, and anterograde transport and retrograde. Especially in knockout, anterograde transport is affected. I'll show you the video here. Please pay attention at the wild type compared with knockout. So in knockout, especially anterograde transport is affected. Retrograde transport is okay. Okay? And here you can see the knockdown of GABA. By knocking down, anterograde transport is also affected, and that can be rescued by the expression of GABA here. Okay, this is the conclusion. So in nerve dendrite, KIF5A bind to the GABARAP and bind to the GABA receptor and transport this cargo. And this is a very important mechanism for the inhibitory synaptic transmission. So if there is a defect in this system, epilepsy is caused. So GABARAP is a very important to suppress or epileptic seizures. So this is one example. Next, I talk about KIF13A, which control anxiety, mood. This is the first motor protein which relate to the psychiatric disease. So we identify KIF13A motor and try to know the function, so we generated knockout mice. So KIF13A is expressed in the cell body and dendrite and also axon. And this is a knockout mice. And in situ hybridizations show that 13A is expressed in hippocampus and in the cortex and other neurons in brain. The knockout mice looks normal at a glimpse, but we study the behavior and locomotions, the crossing, or so this behavior test showed the KIF13A show elevated anxiety phenotypes. So activity is very low. So this is shown clearly by behavior test, by elevated plasmate test. I show you the video. So this test, there is a two chamber, open chamber and dark chamber, the vertical to this plane. So that this is wild type, this is a knockout mice. So wild type mice frequently come to the open field, bright field. But the knockout mice likes dark place. And he's hiding in this place. Sometimes you can see the nose of this guy. Okay? Is definitely is in this box. You will see him. Yeah? Okay. So, but rotor rod tests show that no motor discoordination. No problem about the motor, no motor discoordination. So we thought this could relate to the serotonin system, mood control. First, examine the serotonin secretion. That was not changed. Then we studied the receptor to the serotonin receptor. So this is the staining of 5-HT1A receptor, serotonin 1A receptor, wild type neuron and knockout neuron. As you can see clearly, the surface exposed serotonin 1A receptor was decreasing in the knockout mice. This is the statistics. And so this was confirmed by the effects of KIF5A1A agonist and antagonist on cellular cyclic MP formation in the cultured hippocampal neurons. Because of the sake of the time, I don't go in detail. And this is also specific for the GABA 1A receptor. Okay, so we try to study the mechanism for the transport. And in this case, we found out KIF13A directly bind to the 5-HT1A receptor, serotonin receptor, 1A receptor. And the region, important region, that FHA domain, just after the motor domain. So the motor directly bind to this receptor at the FHA domain. This was confirmed by this expression studies or knockdown studies. So this is the GFP, serotonin 1A receptor, tend to localize on the surface membrane, but this is a knockdown. If you knock down KIF13A, the serotonin receptor tend to accumulate in the cell center. And this phenotype can be rescued by expression of full-length 13A, but cannot be rescued by KIF13A motor domain only, but can be rescued by KIF13A plus FHA domain. This is enough to transport this receptor to the plasma membrane. So that is confirmed by rescue experiment of 13A knockout neuron. This is if you express the full-length 13A, you can rescue the expression of receptor, but motor domain cannot rescue, and if you express motor domain plus FHA domain, then you can rescue this phenotype. And plus, I can show you the video. In wild type, you can see the active transport of 5-HT1A receptor, serotonin 1A receptor from cell center to plasma membrane. But in knockout, the receptor tend to be stay as in cell center, cannot move. Here, in wild type, the receptors incorporated in the tube and move from cell center to plasma membrane. Okay, but in the knockout, they stay in a cytoplasm, cannot move. And this slide shows clearly, this movie clearly shows the wild type, the movement of the serotonin receptor in wild type neuron. It's incorporated in the plasma membrane. Okay, and we can reconstitute the transport of serotonin 1A receptor by KIF13A ATP along microtubule layer in vitro. So this is the conclusion. 13A bind to the serotonin 1A receptor through the FHA domain and transport this receptor from cell center to plasma membrane. And this process is very fundamental for the serotonin neuron function. So if you have a defect in this process, the result is elevated anxiety. So this mice could be good model for the study of the anxiety neurosis or depression. The last motor I would like to introduce is a 19A. This is also very interesting. They control serially length and deletion cause hydrocephalus or female infertility. We identify 19A motor, and I show you this motor moves along microtubules at 0.02 micrometer per second, slow motor. So this is in vitro motility assay. We attach the motor on the cover glass and pour the microtubules on them at ATP. So motor moves on the cover motor attached on the cover glass and move. So the microtubules on the motor can move. So you can see the velocity. Okay, so then this shows the directionality of the motor. This is the seed microtubules, and here is a placent. The tubulin is growing here. And so you can judge the motility of the motor. So as you can see here, the microtubules moving towards minus n. That means this motor moves towards plus n on the microtubules. So to know the function of KIF19A, we knocked out KIF19A. And the first, the phenotype is hydrocephalus. This is wild type. This is the knockout mice. And the ventricular surface is covered by ependymal cell, which has a lot of cilia, okay? And second phenotype is a fallopian tube obstruction. This is wild type, this is mutant. And in the case of mutant, in the lumen of the oviduct is filled with debris or dead cell. So this cannot be cleaned out. So this is the wild type. And all this fallopian tube, oviduct or ependymal cell surface, there are cilia, like this, okay? So we focused on the cilia. So this cilia controls cerebrospinal fluid flow in brain, and in oviduct, they flow eggs, debris, and the mucus in oviduct. So this is the oviduct, a wild type and knockout mice. This is the cilia, and you can see by knocking out 19A, the cilia length increase twice or 3 times, long cilia. Interesting. And that is true in another ciliated epithelium such as trachea, wild type and mutant, and also oviduct, wild type and mutant, wild type and mutant. This is also dose dependent. So I show you the video of the oviduct, wild type and mutant. Look at the ciliary movement, also the movement of the debris in the lumen. In the wild type, ciliary motility is quite oscillated, and so can flow this fluid, so debris can move. But in the knockout, because the cilia is too long, they cannot incorporate the movement, so cannot generate proper flow. So the flow cannot be generated properly. Okay? So summary. KIF98 plus and directed motor and loss of 98 induced hydrocephalus and fallopian tube obstruction and cause female infertility. And knockout mice have abnormally elongated cilia that cannot generate fluid flow. So what is the mechanism for this phenotype? So this is the oviduct epithelium stained with ciliary marker, anti-acetylated tubulin, and 19A antibodies. And this is a cultured ependymal cell. As you can see clearly, 19A tend to accumulate in the ciliary tip. So we isolated the cilia from porcine oviduct, and this is a single cilia, and 19A staining, as you can see clearly, the 19A tend to accumulate at the tip of the cilia. And then we use this in vitro assay. So this is the Chlamydomonas flagella. And if you add brain tubulin polymerize at the plus end and microtubules grow, okay? So if you add KIF98, what happens? So it's a dose dependently, microtubules growing is inhibited, okay? That means KIF98 depolymerize the microtubules ATP dependently. So I show you the in vitro phenomenon. Okay, I stop. This is the microtubule seeds plus N, and if you add 19A, the microtubules depolymerize from the plus N. And biochemistry shows that ATP dependently 19A depolymerized microtubules. So this is the conclusion. So 19A move along microtubules towards plus N at the accumulated ciliary tip, the plus N, the depolymerized microtubules, and control the ciliary length. So this is a very fundamental for the control of the ciliary length. And if you have a defect of this, hydrocephalus occurs, and also female infertility occurs. So as you heard, in neuron in other cell, motor proteins transport various kind of cargo, which is important for the cellular function. But in vivo level, they control higher brain function, or brain development, wiring, or suppression of tumor, or control of the development of peripheral nervous system, and controlling, or very important for the suppression of the diseases such as anxiety, neurosis, or hydrocephalus, female infertility, and epilepsy. So the last slide shows our real key member in laboratory in Japan, and the names that contributed the presentation today was shown in the previous slides. So thank you for audience.
Thank you for your very interesting lecture. In the interest of time, we're going to move to the next talk. The next presentation will be given by Professor Stephen Scher. He's from the Center of Applied Genomics Hospital for Sick Children, University of Toronto, Canada. The title of his presentation is Autism and the Related Neurodevelopmental Disorders, the many genes involved.
Okay, thank you very much. It's very nice to be in a warm spot. When I was traveling to the airport on Saturday evening, we had our first snowstorm in Toronto, so it was nice to come to sunny Saudi Arabia. I work in a pediatric children's hospital in Toronto, and 70% of the admissions to our hospital have some form of genetic disorder. So we're applying gene-based, but now more often than not, whole genome-based assays to try to understand the genetic etiology that contributes to why these children are coming to the hospital. And I just plotted on this slide the massive increase in the number of genome-wide type assays that we're running, both in our research, clinical research, but also in the diagnostic laboratories. And these include not only DNA-based clinical microarrays, RNA gene expression arrays are being used actually more often than not, particularly for cancers. And now more recently doing exome sequencing we heard about this morning. So these are sequencing the 1.6% of the coding regions of the genome. And I'll tell you more about our whole genome sequencing work in autism later in my presentation. So the goal here is really to use these types of tests both in confirmatory diagnoses, prenatal, postnatal, in some cases screening, to try to understand better the genetic factors involved so we can treat the families and manage their disorder in the right way. So I'll tell you about our work on autism spectrum disorders, or ASD. This is a heterogeneous spectrum disorder involving impairments in 3 domains of function, those being social communication deficits, language deficits, restrictive repetitive behaviors. And there's different scales that one can use to measure the degree of severity of these different attributes. In particular, there's 2 diagnostic gold standard scales that we use in assessing all of our patients. And based on the level of function, these children can be grouped into a milder form of ASD called Asperger's syndrome, autistic disorder, childhood disintegrative disorder, and the more severe end, PDD-NOS, or pervasive developmental disorder not otherwise specified. And what I think is very interesting here is that many of the characteristics that we see in autistic children, we see in the general population also. So keep that in mind when I talk about inherited forms of this condition. I'll come back to the prevalence in a moment, but in North America right now, 1 in 88 newborns will develop autism within their lifetime. I think what's really quite important, probably most people are not aware of, that there's many additional coexisting mental and health conditions associated with autism. And these encompass attention deficit, we heard in the last talk, ADHD, anxiety in roughly 14% of cases, sleep disorders, 15% of cases, learning disability or intellectual disability in roughly 25% of cases, depression, anxiety, and then other health conditions, digestive system disorders, brain spinal disorders, and in particular epilepsy. We've seen upwards of 40 to 50% of cases that have autism will also have an epilepsy. So it's really important to understand what the factors involved, because in some cases we believe that the autism is arising as a secondary insult, a secondary effect perhaps due to a brain impairment or damage. In other cases it may be the primary lesion leading to some of these more medical complications that are also associated with the condition. Autism is an urgent public health crisis. As I mentioned, in North America now, and this is in both the United States and Canada, we see a diagnosis in roughly 1 in 88 newborns, and this increases in male because there's a significant gender bias in autism, roughly 4 to 1 male to female gender bias. Statistics from Asia are roughly the same. In fact, some data coming from Korea and Japan indicate that the prevalence is higher, and there's studies ongoing in the Middle East now, including Saudi Arabia, that show that it's probably a uniform condition, indicating that there may not be as much of an environmental contributor, and perhaps genetics is the primary factor involved in autism. So why do we perform research and training in autism? I think the truth will be found in the science. For many years, autism was blamed on the parents for improperly rearing of their children, the so-called refrigerator mother syndrome. And I think, as I'll show you, we've now started to identify some of the genes, some of the real science behind the disorder. Most importantly, earlier detection of autism and related conditions will benefit the health outcomes. So this is the genomic medicine we're here to talk about today. Better understanding of the causes of autism will benefit medical management, including counseling of families, and perhaps also lead to identifying the right pathways that are affected in these individuals so that proper drugs can be used to try to treat them. And then I think very importantly also is to understand human biology, human development. Some of the core features that are affected in developing children who are autistic are cognition, speech, and behavior. And these are the I think, unique features that make us actually human, divide us from the species, lower species. So I think understanding the genes and the proteins involved will also lead to a better understanding of ourselves. In the last 30 years, there's been significant progress unraveling the genetics of autism, and in particular studying identical twins, or twin studies showing that, depending on the study that's been conducted, roughly to 50 to 90% of monozygotic twins, so those twins that have the same genome, will have concordance for the autism spectrum disorder diagnosis. So, and that drops down to 10% if you're looking at dizygotic twins. So this is kind of a hallmark characteristic that genes are indeed involved. There's a roughly 18, there's a newer study that has come out that shows 25% risk to siblings. So a second-born child will be on the spectrum much higher than the typical 1% frequency in the population, again indicating genes are involved. I mentioned the 4 to 1 male to female ratio. And this is something you don't hear a lot about, but at least 20% of the parents, so one parent of an autistic child, will also be on the spectrum. So typically they're subclinical, they have what's called a broader autism phenotype, perhaps Asperger's syndrome, and this leads to indication that quite often perhaps inherited genetic factors are involved. Interestingly, you see variability of clinical presentation within the same family. So in these identical twins, roughly say 80% of the time they'll sit at the same kind of point on the spectrum, but in some cases you'll have one identical twin that's severely affected, and the sibling is actually mildly affected with something like Asperger's syndrome. There's been many genome-wide association studies conducted around the world over the last decade, and all data suggests that there is a very small, if any, role of common genetic variants involved in autism, and the real story resides in rare genetic variants. And as I'll show you, there's no gene that's been identified yet that has more than 1% frequency in the autism population. So there's lots of genes involved, coming back to my title. This is a slide that summarizes what's known about the genetics of autism. Roughly 20% of cases that would come in, say consecutively in our clinic, we can now explain based on some type of genetic explanation. So in roughly 10% of these cases, the child would have an ASD-related syndrome such as Fragile X or tuberous sclerosis. Doing chromosome analysis, so karyotyping would reveal roughly 5% chromosome rearrangement, some of these shown the second bubble here. And then using higher resolution microarrays or clinical microarrays, you would find a de novo or a rare inherited copy number variant affecting regions such as chromosome 16, P11.2, and others in roughly 5%. And more recently, doing targeted sequencing and exome sequencing has revealed another 5% or so of mutations, loss of function damaging mutations in genes, and I'll come back and explain these in a moment. So that adds up depending on how you group it, but we've done these studies now on consecutive cases coming to our clinic, and we can ascribe a genetic role in about 20% of these cases. So if indeed there's 90% heritability, what's happening in the other 70%? And I'll come back to that in a moment. So these are the high penetrant risk genes that have been identified now so far that are screened in molecular diagnostic laboratories. There are many others. These are the top ones that come out in our Canadian cohort. So the deletion, the 600 kilobase deletion or duplication on chromosome 16, deletions of the Norexin gene, PATCH-D1, DDX53, and others listed here. I'll come back and talk about the ones that are highlighted in red. But what I wanted to point out is that the p-value and odds ratios associated here are very high. If you have a significant mutation in one of these genes, you're most likely to fall somewhere on the autism spectrum or have one of these other neurodevelopmental conditions. And I think this is the real story that hasn't gone out yet in the literature, is that we're seeing mutations in these genes in other neurodevelopmental neuropsychiatric conditions, such as schizophrenia, bipolar, ADHD, intellectual disability, epilepsy. It's not surprising given the overlap in the phenotypes. I won't go into great detail. We heard some nice talks earlier. You can knock these genes out in mice, and you can show using some very nice behavioral assays that these mutations lead to similar attributes in mice. Of course, mice don't talk like humans do, so you can't measure that directly. But I think what's interesting here is in almost every case, you have to have a homozygous deletion or a null in these mouse genes to see a detectable phenotype, whereas in human, I just want to emphasize, most of the mutations I'm talking about are heterozygous, okay? So only one of the genes is affected. This is one example of a multi-generation family, a 4-generation family carrying a deletion that's about 64,000 base pairs that knocks out a large proportion of the synaptic scaffolding 1 gene or SHANK1 gene. This was interesting to us because we only saw the expression of phenotype on this autosomal. This is chromosome 19, so it's not one of the gender and the X chromosome. And every male that carries this deletion is either autistic or has a subclinical broader autism phenotype here. The grandfather was not clinically assessed, but stories from the family indicated that he was probably also on the spectrum. We don't see this deletion in any of over 10,000 controls that have been checked, and we've now followed up and found additional mutations in other males and not female carriers. So this is interesting because in SHANK2 and SHANK3, which are also ASD risk genes, we find mutations in both males and females. So we think this has something to do with the unique biology of this protein. We don't know why yet, but we can use this type of approach to at least point to this gene as something we need to look at, effects of gender, perhaps hormone on development. So I wanted to point this out, and I'll come back to this a little bit later. Of course, had we known beforehand, this was all done retrospectively and through a research study about some of these carriers of the mutation. These always pass the deletion from the female, unaffected, into males. You could have this as an early diagnostic test, so the children could be enrolled in behavioral intervention therapies and treated properly, in some cases before the disorder actually showed in the clinic. This is another interesting locus that we found a few years back that's on the X chromosome. So if you have a deletion of this anywhere on this 1 megabase region on the X chromosome, XP22.1, and you're a male, most of these changes are inherited from unaffected females. Again, you'll be somewhere on the autism spectrum or have intellectual disability. And it's difficult to see this, but I'll just quickly show that there's a 3-exon protein coding gene called PATCH-D1 that encodes a protein involved in GLE transcriptional regulation of the hedgehog signaling pathway. So we found mutations in the protein coding gene, but also there's deletions upstream in a non-coding RNA that we think is involved in regulating this protein coding gene here. So lots of deletions identified. Again, these are always in males. Whenever we find a deletion come up in this region in something of a different cohort, like intellectual disability or ADHD, and we go back and rephenotype, we actually find that that individual is on the autism spectrum. So far, this locus, and again, still early days, is autism specific, and I think that's quite interesting. Now, some of you will probably recognize some of these genes. So far, at least most of the bona fide susceptibility genes are all encoding proteins that are acting at the synapse. So the neurexins and neuroligands, the ones that have been identified, are shown in red here. Okay, and I'll come back to this a little bit at the end of my talk. This is just an example showing what's happening structurally. We think that autism probably in most cases, not in all cases, but probably in most cases has to do with a change in homeostasis of the dosage of specific proteins involved in synaptic scaffolding. And this is an example of the SHANK2 gene, where we introduce mutations in a dominant negative type manner, expressing rat hippocampal cells, and could show that the dendritic spine volume was altered when you introduce this type of mutation. So in some cases you have too many spines, in some cases too little. Once you kind of throw an imbalance in this network of spines, it causes problems in neuronal processing. Okay, so how do we find the remaining rare variants, the mutations, and the genes involved in autism? The experiments we've done up to date have been what I call a poor man's approach at looking at the genome. We've done microarray screening, which only scans, say, can only detect deletions down to maybe 100 kilobases or so. We've done exome sequencing. Now it only screens protein coding regions. So we really want to do whole genome sequencing. So we did this pilot study, and we just published this, so I'm going to spend a little bit of time on this study. But this is the rationale for needing to go to whole genome or really complete resolution scanning. We estimate that there's 100 genes involved. We've seen, in some cases, single mutations that are sufficient to cause autism. In other cases, you need combinations of mutations. I don't have a lot of time to talk about this, but the C&V studies and the exome studies are largely yielding right now different genes and loci. And I suspect that plays into the mechanisms involved. There are 119 known ASD risk genes now, and 11 of those actually don't have any exon coverage when you use the typical capture arrays that the vendors are using at the present time, including SHANK2 and the sodium channel gene I'll talk about a little bit later. We estimate that 5% of our Canadian cohort has intergenic or intronic mutations. We published on this. I mentioned PATCH-D1 non-coding RNA. There's mutations intronic in Orexin-1 and antisense genes. And then finally, many of the ASD risk genes are on the X chromosome. This plays into intellectual disability story. And the ratio when you're doing sequencing males, have half copy number, plays into, you have to have good sequencing and good coverage. So most of the, actually all of the published exome sequencing studies did not even look at the X chromosome up until recently. So we did this pilot study. We studied 33 families. We've now extended this number up to 500 families where we've sequenced whole genome, the complete family. I won't go into all the details, but we've used Illumina sequencing in this case, done pretty high coverage sequencing. The average was 35.8x coverage of the entire euchromatic genome. All of these samples had been pre-checked using high resolution microarrays, 2 microarrays at a minimum. So we knew a lot about the copy number variation. So these were idiopathic cases with respect to at least the molecular definition.